Thank you, Jimbo, and welcome to others who have just joined us. Um, as a reminder, if you have any questions during the presentations, please feel free to include them in the chat box and we'll address them at the end of the presentation. So with that, we'll move on to the next portion of the agenda, which will now be talking about the HELM adoption and helmmonomer.org. The speakers for this presentation is again Jimbo Lee and also Jeff Milton. And just to give an introduction for Jeff as well, Jeff is CEO of Agila Labs and former head of data sciences at Arcturus Therapeutics, where he worked on several programs, including mRNA vaccine development for COVID-19. Jeff served as the Director of Functional Genomics and Drug Discovery at Ionis Pharmaceuticals. He led the screening technology group. Prior to this, he worked in the bioinformatics department at GeneTech.
Thank you. Jimbo, are you talking first or am I going?
You go first.
Okay. Glad I asked.
Yeah.
All right, let me go ahead and share my screen.
Mm-hmm.
Okay. So I'm going to talk about monomers and HELM, and I'm going to do it in the context of RNA therapeutics, and that's what La Jolla Labs does. It's an RNA therapeutics company focusing on screening technology first. And so I'm going to talk about RNA targeting therapeutics. And these are modalities like antisense oligos or siRNA, like companies like Alnylam in your area. And then I'm going to talk about HELM and monomers in the context of mRNA. So there's just two modalities in this, or two approaches that I'll be talking about. The first thing I want to mention is, why we do this. We do this because there's a need, and there are patients who need these types or need therapeutics, and spinal muscular atrophy is a good example of where there's been a pretty significant success. It is the #1 genetic cause of deaths in infants, and there's actually a solution with this molecule here. It's a very simple molecule. It's a DNA. It's basically a DNA molecule, 18-mer, that has been chemically modified at two positions, two monomers. So instead of a phosphate group, it uses a phosphorothioate, and then there's a modification. It's 2' methoxyethyl modification on the sugar. And this, by all definitions, is a success. Here's an example of a kid, 15, I forgot how old he is, but anyway, Alexander was taking Spinraza in 2017. It's stopped the disease in its tracks. A couple of months ago, or last month, he started school and he was able to continue and thrive. And so while it doesn't reverse the degeneration, it will stop it very effectively. And so that's what it's done, and proven that many times over. This drug is a long, any sort of drug like this, it takes a long time to develop. You've got the academic sort of enterprise here back in 1995, where they discovered the gene that causes it. They discovered a paralog gene that they decided, well, this could be a solution, and I'll get to that in a little bit. And then there's this gap here where they actually take this idea, these concepts, and turn it into a physical reality, a real drug, and a mechanism of action. And then this long line here is the clinical part of it, and the preclinical and clinical studies. Oh, by the way, my slides are labeled confidential, but they're not confidential. You can, I just didn't take that off. So when we look at the mechanism of action for this particular drug, you have to understand that there's one gene that basically is ineffective, completely useless, and that's this SMN1 gene. And it's a homozygous deletion of this SMN1. But since you have this paralog, which is almost identical in sequence, the brilliance of Adrian Craner and his group at Cold Spring Harbor was that, we can rescue the phenotype, we can rescue this protein just by modifying the splicing so that we have the identical messenger RNA of the SMN1 gene. And so how do we modify that splicing? Well, they decided to run a very large experiment called a drug screening experiment. And this is kind of what it looks like. Each one of these blue dots are oligonucleotides that are that DNA molecule that I showed before. And they screened across the sequence space, and then they screened in the chemical space, and the chemistry modification space. And so Spinraza pops out somewhere over here, and this is an actual reflection of what the actual data looks like. I took this from public data that Adrian Craner published. And so Spinraza falls out here. It causes that exon 7 to be included, which creates a functional and longer half-life protein, which then rescues these patients. This whole process of designing or developing drugs is not a linear process. So you go through, you find you do your primary screen, you do your in silico analysis, and then you do some in vitro you do some in vivo tox studies, and then maybe you want to go back, modify the chemistry based on those tox parameters. And this is where monomers and organizing those monomers becomes super important. So what I'm basically describing is, for this very large experiment, is that you're going to discover, or you're going to do an experiment with respect to sequence, and then you're going to do an experiment with respect to structure. And so what that may look like in the HELM world or the informatics world is something like this, where you see these biopolymers, these are oligonucleotides, and you may want to move this phosphorothioate down the backbone because maybe you want to create an SAR, a structure activity relationship, where you have an idea of where something is more efficacious or less toxic, and you're just tweaking these monomers. And so one of the things, getting to what we have at La Jolla Labs, having this monomer database is super important, because we essentially we design these very large experiments. So this is a screenshot of our software. And we create these templates, which are basically monomer templates that sit on our database. We have our monomer database in the background. And we create these templates, and then we do our screen, which puts the sequence into those templates and creates full covalently bound molecules that you can then export. And then we basically creates this type of structure. And this is a Silogence software for visualizing some of the compounds that we create. And so once you have that, then you can do, actually, I'm going to jump back a little bit and just go over some of the SAR stuff. So if we have our sequences tiled across these, in this particular case, it's these ASOs that are tiled across the gene. And then we actually have a software tool that we go through, and we can do our modifications, for example, the phosphorothioates across the backbone for a particular structure. So you'll see we have tile across the sequence, and then we have a pile up for structure. And then we print that out into an Excel doc and send that off to IDT. And here's the key, and this doesn't exist yet, but would be super powerful, is if IDT had the monomer database, had access to this system of record, this global system of record of monomers, and then all we had to do was send over our biopolymer identifiers, and they would know exactly what that structure looks like, complete, the oligo structure. So that doesn't exist today, but that's the push. It's a slow moving thing. We're getting there. And especially with Silence's help, they've been instrumental in helping us push that. But so this is an example of what that structure looks like. So this is actually a different example, but these are siRNAs. These are actually clinical drugs. These are drugs that were in the clinic. And in fact, I think the Quark one is not anymore, but it was in phase 2. But each one of these identifiers are pointers to a monomer database. And if they're common monomers, they all exist as they're curated as something that's a global reference. Now they may be proprietary, and that's when you have your own database, and I'll show that in a little bit. But in any case, what you end up with is in our software, we end up with this big screen. We actually run the experiments, and then we end up with this huge heat map of where we have structures that worked, compounds that worked, and compounds that didn't. And the cool thing is, because it's in HELM and because it's backed by a monomer database, we can search based on the biopolymer structure. So I can say, give me the top ones with the phosphorothioate backbone in these locations. And then I can also search, because it actually references chemical structure. I can go back and actually search for, give me the aromatic, give me the structures with the most aromatic links, that type of thing. And so that's very powerful. And I'm going to jump very quickly into the mRNA. I'm going to skip through this because I don't want to take too much time. I know, Jimbo, you're going to talk as well. So I'll skip through this quickly. But the mRNA structures are also modified in many cases, not always, but some. So you'll find that there's a higher level structure. So there's there's the cap, the 5' cap, the 5' UTR, there's the gene, there's a 3' UTR and a poly-A tail. And so you'll have modifications in here. These are the actual structures of the Pfizer and Moderna mRNAs. And so what they do is basically these highlighted areas are those different the UTRs and the main protein. But there are also different ways of representing mRNA. And so the common one you think of is this, where you have the cap, the UTR, and the coding sequence. And then there's also another modality, which actually includes a replicase. And so these are the higher level non-monomer, but these are sort of the annotations that are associated with these mRNAs. They're things that need to be captured in software. Another part is that you can't separate out the idea that there is a lipid nanoparticle that goes along with this, because that is actually a very important part of the driving factor of whether or not it works, is how well that lipid nanoparticle works. So you have to kind of co-mingle these things, and it gets very, very complicated. And I already sort of covered this a little bit, but for the most, so what my point is basically that for HELM or for mRNA, HELM doesn't really work, but monomers do. And so there are things, HELM has to evolve a little bit more. You have large structures, and it's a nice way of representing the structures, but it doesn't capture things like you may want to have some secondary structure captured, which is kind of important for some of the assays that if you're looking for specific activity of a drug, and you want to be able to compare whether or not there are secondary structures in different locations. That's something that needs to be sort of worked out. Also, things called codon optimization. These are things that need to be captured in HELM as well. But in any case, so this is my sort of final slide. And what I want to just kind of point out is that the monomer database is the system of record in our world. And this is actually what I built at Ionis as well. So, but this highlights the flow of information for how we're building our software tools, and how really anybody who's doing these types of biopolymer software tools should work off of. We have a system of record, which is helmmonomer.org. And Jimbo, I will say that it's probably going to be monomer.org very soon here for you. We're working that out. And then you have your own database. But the key here is that you're pulling in the system of record, the global reference database, into your instance of your proprietary structures. So you're having them commingle, but using this reference database. And then your biopolymer database is just pointers to those monomer database. So you have an editor, and that's the Silogens tools are going to be providing those sort of the front end to the monomer database. your monomer database, and then well, for us, our screening tools are going to be using the biopolymer database. And so I'll end there, but that's just kind of a quick use case example of how it works in our therapeutic world.
Thank you, Jeff. I think you make my presentation much easier to understand.
Glad I could help.
Great. So, with Jeff's, laying out the story, the needs of HELM technology and actually how HELM technology should work. And so I'm going to share a little bit about really, there's what kind of hurdle actually to a organization or a company or academia institution to actually adopt and implement HELM. So I'm going to share my screen. Great. I'm going to skip all the intro slides. Okay, and there are actually mainly three main hurdles for HELM adoption and HELM implementation. As Jeff mentioned, one of the hurdle is really when HELM first released to Pistorius Alliance and to the scientific community, is the user interface was a thick client Java-based app, so require local installation for each computer that user use to access that technology. So that hurdle has been addressed and has been resolved with the first release of the HELM web editor in 2016. And then the second and third one is a little bit more challenging especially for small organizations and also academia, research group. And what that is really the adoption or implementation does require IT infrastructure and knowledge. Not every organization have someone like Jeff, so can easily put together a database and actually write specialized software as well. So that is another hurdle to kind of slow down the adoptions, especially in a wider scientific community. And the third one is what Jeff mentioned. You do need the monomer library to start in order to put together biopolymer. Those are the building block for the biopolymer. So how do you actually have access to a monomer library and make sure those monomer library are well curated and without a lot of error in it, and also is well maintained, because a monomer, especially in our organization or in the scientific community, that monomers evolve over time. There's always novel monomer created by different organizations or different research institute, create over the time. So how do you can have access to a well-maintained monomer library, not just one, have access to all the monomer library that is really help you to facilitate your research. That is another challenge. So how monomer.org or the monomer.org platform is actually conceived by Jeff and other colleagues, heavily involved in the HELM technology development. And the purpose of that, let me just give you one more example. I think Jeff already mentioned, right? If Laureate Biotech actually has a specialized proprietary monomer library, but that is not actually easily accessible, or they are not comfortable provide access to the external research community, then each organization may have different monomer definitions. And this is a very simple example. So let's say this is a so-called methylcysteine monomer of amino acids. In company one, the structure of the MEC is actually the left structures. In company two, it's also called MEC, but the methylation is actually not on the N terminal, it's actually on the cysteine side chain in the sulfur, right? And then in the company three, it's actually the same structure on the left, but it's called MC instead of MEC. So if you have a HELM string, if you don't have the access to the correct monomer library, your interpretation of this HELM structure or HELM string could be incorrect. So that is really a very common challenge. It's almost not practical for the whole scientific community in the world using a single monomer library, because those as I mentioned, those monomer, the library composition are evolving over time and very rapidly as well. So how can we solve this challenge? Okay, basically is you know, it's kind of a, you know, many to one and one to many type of you know, a problem. Okay, the solution is actually what we developed here over the years. It's called the HELM monomer.org. Okay, the purpose, it serves two purposes. So this scientific platform, open to everyone in the scientific community, will serve as a online dictionary for HELM monomer. So it's a web portal, and then I use crowdsourcing monomer, okay, and build up monomer library. And it's also a platform to securely manage and maintain monomer libraries. Monomer library can be private or public. So for a company like La Jolla Biotech and like Moderna and maybe Alnylam, they can actually also use this platform to maintain their, you know, private library. They don't make it public. So you know, other company, maybe a general audience, we will not have access to their private monomer library, but they can actually allow access to their private you know, library to certain collaborator of theirs. Okay, and the key word here is securely manage your monomer library. Okay, and the second purpose of the HELM monomer.org is actually as a translator, as an interpreter of HELM structure or HELM string. So user don't need to have a login into the platform, and they can access the so-called public monomer library. Okay, and they can use that to create HELM structure or HELM string. And they can also use that to translate HELM string. So for example, if I'm looking at publications, right, and it could be, you know, you can access that through PubChem, for example. If you find a HELM string, and now you can use this particular platform to kind of translate that HELM string. All you need to do is actually find the library, monomer library, used to actually create a HELM string, and then you do the translation. So that then you're confident that the translation will be correct because you are using the correct monomer library. And what does the HELM monomer.org looks like? Okay, I'm going to share a few screenshots with you. So it will be this is the landing page. Basically, here is, you know, user don't need to have an account. They just come here, select the monomer library available, which is the public library. They can paste in the HELM string, and then this platform going to do the translation for you. You know, based on the HELM string, and for example here on the left, this is the HELM string I paste into the landing page, and this is the result that returned. And I select the monomer library. This is the HELM core library, which is put together by a dedicated team of a HELM Pistoia enthusiasts. So basically, you know, this you can do the translation and then also provide you some basic calculated property of this particular HELM string as well. Okay. So in addition, as I mentioned, you can actually use this platform, select your library, actually to sketch your HELM structures and also create your own HELM string. So what you may notice that the HELM string created here actually have a destination of the library use. You can see here, this particular HELM string has been using HELM core library version 3. So library actually have version control in this HELM monomer.org platform. Okay, so in the future, when you paste this HELM string in the translator on the landing page, the system, can actually automatically detect what library has been used to create this HELM string. So, so that will be in the future version release of the HELM monomer.org. Okay, and if you are an organization that would like to use this platform to securely manage your monomer library, then you do need an account and you do need a login for security purpose. And this is the login page for user that will have the ability to manage monomer library and maintain monomer library. And so what that monomer library management user interface look like? It's like this. And you can have multiple library in your account. And then the library will be version control. Every time you introduce new member to the library or delete certain member of the library, a new version will be created and will be tracked in the database. So you can actually select previous version of the HELM monomer library to create your HELM string or HELM structures. Okay. So that's what I would like to share with you about HELM-monomer.org. The target release day of HELM-monomer.org will be November 15th. It's a little bit 2 weeks away. Yeah. I will stop my presentation here. Any questions for Jeff and me?
We do have a question from the chat room.
Mhm.
And it is from Steve Heller, and it is, what is your relationship between your HELM activities and those of IUPAC?
Okay, excellent questions. So IUPAC has been, I think Pistoia Alliance has transferred the HELM, I would say the operation to IUPAC. So IUPAC is going to take the lead on put together so-called IUPAC, right? The rule for how to name a monomer. So IUPAC will take the lead of actually get together, organization who's going to use HELM technology. Now set up the so-called IUPAC rule for how to actually naming of a HELM monomer. And HELM-monomer.org, this is a software platform. So basically, we don't do any setting rule for how to name the monomer, but we provide a scientific online platform for scientific community to actually have access to those of so-called HELM core monomer from IUPAC, and previously is from Pistoia Alliance. So we provide a secure platform to make HELM adoptions a reality to a wider scientific community. Yeah. Does that answer your question, Steve?
Yeah, I want to follow up one with that. The original structures that are in the reference database were actually used. There was a rule document, a nomenclature document that was written up by a guy named Eric Swayze, who was at Ionis Pharmaceuticals, and he curated the first data set of that reference data set. So he tried to model the naming structures that he's tried to follow the IUPAC rules for how to name these structures. So there's an overlap there from the beginning. But now that it's moving to IUPAC, it's not that there's a lot of uncertainty around how this is going to work out, because obviously these are they're sort of driven by committee, so things could be slow. So it's one thing to the great service here that I think Silogens is doing is it's providing you with the ability to evolve your monomers outside of any sort of rate limiting step, like a large committee naming structures. You can basically use that reference data set that's there right now, and then start just building your own data and then merge later.
Yeah, I will echo Jeff that this platform allows you to manage versioning of the monomer library too. So as the core monomer set get evolved over time, you may end up in different version of that. So you can always refer back to the older versions using this platform. Yeah. Okay. Any other questions before we wrap up this talk? Okay, if not, let's move on to the next talk. I'm going to stop sharing.